you a unique opportunity to start understanding the regulatory code of the genome. So as you know, regulatory elements, they are bound by a variety of different transcription factors. These TFs often bind in a highly combinatorial fashion and often have strong cooperative effects on each other. And what's interesting is there's a lot of information about regulation encoded in the genome sequence itself. So transcription factors, as you know, recognize motif sequences. And what we're really interested in is not just discovering these motifs, and there's been a lot of work in this space, but we are more interested in trying to figure out sort of the higher order syntax. And what I mean by syntax is obviously the composition of these elements, that is the variety of different combinations of motifs that exist, but also other kinds of rules of arrangement, such as preferred spacing, orientation, density, and the effects of this syntax on TF cooperative binding. That's really what we're interested in. So I'm going to jump right in and sort of try to explain how you can model or try to address this question as a machine learning problem. So if you're given, let's say, a ChIP-seq experiment in the genome, you get a readout across 3 billion base pairs in some particular cell type or context of interest. And what the typical approach is to take the genome and you can bin it into various small size sequences, let's say 1,000 base pairs, 100 base pairs. And so what you end up getting is you end up getting millions of little DNA sequences of some length, depending on your binning of the genome. And each of those sequences is mapped by the experiment to some real valued number corresponding to the intensity or the enrichment score. Or if you decide to threshold these profiles, you effectively get a binary label telling you if the sequence is likely bound or not. So this is a classic supervised learning problem. We are given a collection of input sequences, and the goal is to try to learn a model that can map these sequences to continuous or binary labels. And a variety of different machine learning methods have been developed to solve this question. So we turn to neural networks for the primary reason that, of course, they are very powerful predictive models, but more interestingly, from our perspective, since they're able to learn features from raw inputs and map those very accurately to outputs, we're actually more interested in the representations that these models actually learn. So I'm just going to give, I'm sure, I mean, this is a machine learning audience. So everyone's probably pretty familiar with neural networks, but I'm just going to give you one slide introduction. So our primary goal is to, in this case, learn a mapping from an input DNA sequence to an output label. The output label could be binary or continuous. In this case, let's assume it's binary for now. So we take these sequences, we represent them as one-hot encoded sequences. What that means is a matrix of 4 rows and L columns. L is the length of the sequence. The 4 rows correspond to ACGT, and you put a 1 at the, for the letter that's present at each position in the sequence. Okay. And the goal is to map this one-hot encoded sequence to binary or continuous output. And so the basic building block of a neural network is an artificial neuron, which is basically a simple pattern detector. So it takes some inputs X and produces an output Y. In this case, this neuron is observing or operating on 5 nucleotides, which have been represented in this one-hot encoding. So that's basically 20 inputs, 5 by 4 positions. And the simplest type of function you can think of is a linear combination of these inputs, 20 inputs to produce some output, right? And so if you visualize these weights of this linear model in the same configuration as the inputs, you basically get a matrix of 5 positions by 4 rows and a bunch of weights corresponding to positive or negative numbers. You can visualize this, and as you will notice, it's basically nicely representing something that's reminiscent of a position weight matrix. So basically the weights of an artificial neuron operating on a sequence effectively can act like a motif detector. And so in this particular case, let's say we have this hypothetical motif. We use it to score this piece of the sequence using this simple linear combination. we get a score of 11.6, telling us that this motif this input sequence is a very good match to the weights encoded by this neuron. And then, of course, you can take this linear combination and push it through a nonlinearity. A commonly used nonlinearity for predicting binary outputs is a logistic or a sigmoid, which just squashes this linear output in the range of 0 to 1. In this case, you can see it squashes it to 0.996. Or you can use a rectified linear unit, which is a simple thresholding rule. If the linear output of this neuron is less than 0, you set it to 0. If it's greater than 0, you keep it as it is. So basically, the neural network, the components of the neural network act as these pattern detectors, learning motif-like patterns. And so if our goal is to, again, take this sequence and map it to these binary continuous outputs, obviously, DNA sequences can contain more than one motif sequence. And so we'd want multiple pattern detectors, not just one. So in this case, we have 3 neurons, each of which is encoding different weights, capturing different patterns. And just as you take a motif and you scan a sequence, you can take these multiple pattern detectors and scan the sequence. And this operation is known as a convolution. And that's how you come up with a convolutional neural network. The outputs of this first layer of convolutional neurons, or pattern detectors, can then be fed into additional layers of pattern detectors. And those additional layers learn higher order combinations of these motifs. And ultimately, the last layer of the model is trying to predict a binary continuous output. So it can be a simple linear regression if your goal is to predict an output, or it can be a logistic-like model to predict a binary value. So these kinds of models are quite popular in the literature. They've been introduced since 2015, and there's been a lot of interesting work in this space. You typically train these models on the whole genome, on a bunch of training chromosomes, and you optimize the weights or the coefficients of all of these patterns to minimize a loss function over the training example. So that could be a mean squared error loss if it's a continuous output, a Poisson loss if it's counts, or it could be a logistic loss if it's binary labels. So that's the classical kind of models that you see in the literature. Now, I'll try to argue that these models potentially lose a lot of information. And the reason is because if you take any of these assays, ChIP-seq, or let's say we take a higher resolution version of these, Chip-exo, which I'll introduce in a second, or even DNA-seq and ATAC-seq, and you observe the distribution of reads on the plus and minus strand from any of these assays, you'll notice that there's very intricate base resolution profiles, sometimes stranded, sometimes unstranded. These are often referred to as footprints in the case of DNAs and ATAC-seq. And if you just summarize this information in a large sort of low resolution window, let's say of 100 or 200 base pairs, you lose this kind of really nice information about where the protein might be contacting DNA and the precise sort of effect it has on the read profiles around the binding position. So our goal is to try to leverage this more fine scale structure into the modeling. And so we collaborated with Julia because she has invented basically an efficient version of the Chip-exo experimental protocol. The basic idea behind it is similar to Chip-seq, where you pull down sequences bound by a TF of interest using an antibody and sequence it. In this case, you use an exonuclease, which actually chops the DNA all the way up to where the protein contacts DNA. So you get much more high resolution footprints as shown here, and they are stranded. So you get reads on the plus strand, reads on the minus strand, precisely sort of bookmarking the binding site. And so these are really very accurate, very precise base resolution experiments. And so we really want to leverage these kinds of data to really see if our models can improve what we can pull out of these kinds of profiles. So we're going to focus on 4 transcription factors, Oct4, SOX2, Nanog, and KLF, which, as you all know, are classic pluripotency transcription factors. And we're going to study this in these data sets in mouse embryonic stem cells. And there's a good reason we picked these TFs because they're actually very well studied. And so part of the challenge we wanted to pose ourselves is Firstly, we can use models built on these TFs to recapture a lot of existing information that we know about these transcription factors. And secondly, maybe we want to see if we can learn something new, even on very well-studied transcription factors using these kinds of models. So we adapted the classical approach of taking DNA sequences and mapping them to individual, like a single scalar label, which could be a binary or continuous value, to a model that takes in sequence and directly maps it to base resolution profiles on the plus and minus strand. Okay. So our goal is really to take in one hot encoded input sequence, 1 kilobase long usually, and map it to these base resolution profiles in the plus and minus strand. And we have several novel aspects in the model itself. We call this model BPNet, or base pair resolution network. So first thing is that we wanted to model these profiles in a sort of a different way. And we came up with a novel loss function. The part of the idea is we wanted to decouple two properties of these profiles. One is the total read counts in this 1,000 base pair window, right? This 1,000 base pair sequence. So you want to model effectively the total magnitude of the signal in the entire sequence, across the entire sequence. And then you want to model how those total counts are distributed across each base in the sequence. So we have two loss functions. We have basically the loss function is a combination of two individual losses. The first loss function is the mean squared error, which captures the error in the log of the total counts across the entire sequence. And the second is a multinomial negative log likelihood, which really nicely captures how reads are distributed across each position in the genome. So it captures really the positional distribution of the total counts across 1,000 base pairs. And this really captures the noise properties of, in many cases, very sparsely sampled regions quite nicely, and allows a model to predict an expected value, thereby denoising the signal. There are a few other innovations we have here in this model. Our model allows for automatic bias correction. So what we do is we provide a bias track. In this case, for example, for Chip-exo, we use something called a patch cap input. But for ChIP-seq, for example, you can imagine using input DNA sequence input DNA, a wild-type control. For attack-seq and DNA-seq, you can imagine using TN5 or DNA's bias. Basically, we feed these bias tracks as auxiliary inputs to the final layer of the model, and the model learns to automatically regress out the effects of these bias tracks and then fit the residual to the sequence. Okay, so we don't have to do explicit bias correction. The model learns to do an automatic bias correction. In terms of the architecture, there is nothing particularly special. The main thing is it's a fully convolutional architecture, so we avoid all kinds of any kind of pooling operations as far as possible. And in terms of the convolutional layers, we use a combination of regular convolution in the first layer, followed by dilated convolutions with residual connections, allowing us to build reasonably deep models that have sufficiently large receptor fields. Okay. So if we take these models and try to see how they actually work in terms of predictive performance, so what we do is we train the models on a subset of the genome, so a subset of chromosomes, and we test on the remaining. And we also learn, I forgot to mention one thing, we train multitask on all the 4 transcription factors. So we're jointly training the model on all 4 TFs. So the model can effectively share parameters across the 4 TFs, particularly because they really like to bind with each other. And so we can borrow power from each of the data sets. So I want to show you some examples, start with some specific loci in the genome to just show you how the model is able to make predictions on chromosomes not seen in training. So this is one locus in the genome. Here you see the Oct4 observed profile. This is the profile predicted by the model. This is for Sox2, this is for Nanog, this is for KLF. You can see in cases where the signal is very sparse, like in this case, the KLF data, the model actually kind of effectively denoises or does certain imputation, when the signal is pretty strong with sufficient coverage, the model is extremely accurate, very similar to what you observe in the observed data. This is at base resolution. This is another enhancer next to the SAL1 gene. So if you look at individual examples, you really, you can, we provide browser sessions as part of the paper. You can scroll around and really see that they are incredibly accurate at base resolution. But obviously we need to quantify this genome-wide. And so one of the approaches we took, these profiles are not continuous. As you can see, they're basically a bunch of noise and then these spikes. So you don't want to use something like a Pearson correlation or anything of that sort. So you wanted to have a profile similarity metric. And so one simple approach we used is we took these profiles and then we binarized them by position to have positive labels on exact nucleotides that match the spikes of the footprints, and all the other positions are labeled as negatives. Okay, so the goal is to really evaluate the accuracy of the model in terms of the positional precision to capture the precise locations of the footprint spikes. And so this is basically effectively becomes a binary classification only at evaluation time. We're not really training on this. We're just doing it at evaluation. And so we can compute basically an area under the precision recall curve, because as you can see, most of the positions are going to be negatives and there are going to be a handful of spikes, positive examples. So it's a highly unbalanced classification task. So you'll notice that we evaluated this AUPRC at different resolutions. So one means single base pair. So that means the spike has to be, predicted spike has to be exactly aligned with the observed spike. But you can allow for some flexibility. And so we allowed for like 2 base pair flexibility, 5 base pairs, or 10 base pairs. So that's about we allow some wiggle room. And we are plotting the AUPRC as a function of that wiggle room. So the blue curve is what you see for the model. And this is again on haired out chromosomes. So sequences sequences that have not been used in training. Obviously, we need some kind of baseline to compare against. So we try to generate various kinds of upper bounds and lower bounds on what the performance could be. So one approach is if you just take the profiles in the training set, so you don't use a model, you just use the profiles in the training set, you take the average profile and you use that to predict the positions, and that gives you the green profile. So that's kind of one kind of lower bound or baseline model. The other is if you just randomize the profiles, what do they look like? That's obviously a lower bound in terms of how poorly you could do, completely random model. And finally, is an upper bound, or trying to figure out what is close to an upper bound. We took replicate experiments for the same TF, and we used basically one replicate to predict the other. We computed the AUPRC that way. And what you see is, at least in terms of footprint precision, that is the positional precision of the footprints, the BPNet model actually is as good, or in some cases slightly better than the replicates, because it's able to denoise the data, and we're evaluating against the pooled sample across the 2 replicates. So we're pretty happy that the models are able to at least perform positional precision of footprint calling at base resolution with the accuracy or concordance that you observe between replicate experiments. So this is, of course, just a measure of positional precision or similarity of the shapes of the profiles. You could use other metrics too, like Jensen-Shannon distance and so forth, which we've done. But an equally important prediction is also the total counts, right? So remember, we have 2 loss functions, one operating on the total counts and the other operating on the profiles, the shapes of the profiles. So the first evaluation metric tells us that we're pretty good at the shapes of the profiles. When we compare the observed and predicted counts, total counts, we see that our models are pretty good, but nowhere close to the replicates, right? So replicates have very high concordance, close to 0.97, 0.96. Models are good, but nowhere close to them. And I personally don't think this is an issue with the models per se. Remember that we're trying to predict binding entirely from local proximal sequence. In my view, it is actually impossible to predict total counts particularly. using a local sequence alone, because you're going to have a lot of effects of local chromatin state, prior chromatin state, as well as effects of long-range or distal interactions. For example, there's lots of evidence of distal QTLs, SNPs, and other locations, distal locations in the genome that affect the magnitude of binding at other locations in the genome. So we're not modeling any of those aspects, so we should not expect to have perfect, very strong accuracy. But this is really nice because it showcases that the model is now able to, using these two different outputs or loss functions, the model can now, we can disambiguate the effects of sequence on the shapes of the profiles, the local footprinting shapes, which are really determined by the local binding events and the local sequence, and the total counts, which is a combination of local sequence, local state, chromatin state, and potentially distal effects. And so we can decouple these two effects and really focus on one or the other. All right, so we have the models. They're making pretty decent predictions. In many ways, the predictions are pointless because we already have the data, right? So the main point of trying to make these evaluations is to make sure that we have a model that is as accurate as possible in terms of making predictions of sequences that it has not been trained on in the same cellular context. So we're not really addressing the issue of taking this model and predicting binding in a new cell type. That's a different problem. And of course, it cannot be solved by a sequence-only model. Our goal is to really fit a sequence model to binding in this cell type and now start dissecting how the model is able to make those predictions. And by having a model that's extremely accurate on predicting binding on sequences in the same cell type it was trained on, you can now start using the model to do many interesting in silico experiments, as I will show you. So we'll try to interpret these models in various ways. And so we'll start off by trying to decipher the predictor motifs and motif instances in these regions. So the way we do that is, now we have this model which takes in sequences and predicts profiles with pretty high accuracy. And so what we can do is use a method, efficient method called deep lift, developed by Avanti in my lab. It's a efficient backpropagation approach, which takes the output of the model and then recursively decomposes the contributions of the neurons across the layers, all the way back down to the input. And so what you end up getting is a contribution score for every nucleotide in the sequence. And it's a simple additive decomposition of the output with respect to some reference. Now, what's interesting is obviously we can do this for every position in the profile. And in fact, that's what we do. So we compute the contribution of every nucleotide to the output, predicted output at every position in the profile. And then what we basically do is we compute an expected value. We integrate over the profile. So we get a weighted combination of the contribution of each base to each position, thereby giving us one contribution score profile for the entire output. Okay. And this has a nice advantage of basically denoising the contribution scores, because if you just have a single output, you can have quite a bit of uncertainty in terms of these values. And by integrating over the profile, which is 1,000 base pairs long, you're basically averaging these contribution scores and getting much more stable scores. And you'll see this in the next talk from Alex from my lab about the relative stability of these kinds of profile models versus classic binary models for interpretation. So yeah, Deep Lift was published in 2017. I think it's still quite competitive in terms of as an attribution scoring method, and the code is available and you should feel free to use it. So what does these actually look like? So we can now take any enhancer in the genome or any regulatory element. In this case, we're looking at the distal enhancer for the Oct4 gene. And we see that there's really nice combinatorial binding of all 4 transcription factors. We can interpret the input sequence with respect to each of these output tasks. And so we get deep lift contribution scores for each nucleotide for all 4 tasks. And we see that we can really map with high precision all the different combinations of motifs that contribute to binding of the 4 factors. And you can see that even though it's the same sequence, each of these TFs uses the sequences in different ways. to bind. So for example, you see ZIC3 contributing most strongly to Nanog binding, and a bunch of these other motifs popping up as important for specific transcription factors. So in this way, we can interpret any single element in the genome, transcription factor binding site. But we also want to summarize these patterns across the whole genome. And so we use a method called TF-MODISCO, which was also developed by Avanti. And what it does is it takes each of these binding sites. Let's say for Oct4, there are, let's say, 20,000 or 50,000 regions and peaks that the TF is bound to. We interpret those regions using deep lift through the model, through the lens of the model. And that gives us these contribution score profiles. Okay. What we then do is identify regions with significant scores. We call these seclets. And then we cluster them based on similarity of sequence and importance of the nucleotides. And then we collapse them into consolidated motifs. And we derive a new motif representation called a contribution weight matrix. It's similar in conceptually to a position weight matrix. That is, for each base, but for each base, instead of summarizing the frequency of nucleotides, which is what a classic position frequency weight matrix does, we instead compute the average contribution scores. Okay, so you can represent it exactly like a motif logo, but the heights of these nucleotides are capturing the average contribution scores. Okay, and I will flip back and forth between the PFM and the CWM representation and show you some of the advantages of the CWM representation. So we run this for each of the 4 transcription factors. And we end up with actually getting about 50 consolidated motifs for just the 4 transcription factors. I'm obviously not going to go through all 50 of them. There are slight variations of several of the classically known motifs. I'll focus on these specific motifs. So we capture many of the well-known ones. There's the Oct-Sox heterodimer, there's the Oct4 monomer, there's the Oct-Oct homodimer, Sox2 monomer. I'll get to these Nanog motifs in a second. We learn slightly different versions of the Nanog motif. We have different versions of the KLF4 motifs. And we learn a variety of motifs of other factors which are not actually assayed. So they are likely representing indirect tethered binding. And I'll address that in a few slides. Now, what's nice is for each of these motifs, we can also compute the average contribution of each of these motifs to each of the 4 transcription factors. And you'll see some interesting patterns, like the Oct-Sox motif has very strong influence on Oct4 binding, Sox2 binding, and Nanog binding, to a lesser extent on KLF4. And you'll see corresponding footprints verifying that, in fact, there is Oct4. Each of these TFs are binding at these motifs, as you can see from the exo profiles. So you see these really beautiful sort of combinatorial combinations of direct and indirect binding across all of these motifs. It's worth noting that you need quite a bit of representation power to capture fully the 4 transcription factors. It's not just 4 motifs, as you typically might think. If you go to several databases, typically you'll get, mostly people use 1 motif to represent a transcription factor. It's clear that in a combinatorial setting, you might require more representation power. So we can map motif instances across the genome very accurately, and we can also sort of, in some sense, summarize the density of how many motifs you typically have in a region. And remember, these are not just motifs, these are not just sequence matches, these are motif matches that actually have predictive importance. So in some sense, they are a filtered version of what you just get from a PWM scan across the genome. In the paper, we have very careful comparisons to how the model's motif calling approach compares to using a classical PWM scanning method. And I'll just summarize and say that it substantially outperforms them in many ways. So I'm going to go into some interesting case studies now. So the first thing, obviously, we see direct binding motifs of all of these transcription factors, Oct, Sox, Nanog, and KLF. But we also see some really interesting tethered indirect binding events. So one such example is ZIG3. We find a motif for ZIG3, and it particularly has a strong influence on Nanog and KLF4. And you'll notice that whenever we see tethered binding, that is, we see a motif that typically the TF doesn't directly bind to, you notice that the footprints also tend to be more fuzzy. So you get very, very clean, strong footprints when you have direct binding, like here, Nanog to Nanog. But if Nanog's binding to indirectly through ZIG3, you typically see much more fuzzy footprints. And we've actually quantified this, and across the board, you see a very nice relationship between the punctateness and the strength of the footprint versus the fuzziness capturing direct versus tethered. And just to validate this, that ZIG3 is in fact bound to these sites, we found Chip-exos, and we found direct, clean, punctate footprint, and so nice evidence that now another cool example of tethered binding is the TF3B box. So this was a strange one because we were like, what is happening exactly here? The TF3B box is typically found next to tRNA genes. And what we found is there's pretty strong evidence that Oct4, in fact, is these TF3B box genes that are lighting up are precisely at a, around 280 highly conserved tRNA genes. And the B box motif is palindromic. So what we do is we computationally orient it with respect to the transcription direction. And what we see is Oct4, in fact, is being recruited potentially by TF3C at the start and at the end of the tRNA genes through potentially what looks like a mini loop of sorts. So the exact mechanism is not clear, but it's pretty clear that Oct4 is binding through a more fuzzy, larger footprint through indirect recruitment at the B box. Now, for Nanog, we found, this was very interesting, we found 3 variations of the motif, and these are pretty distinct. They share a core TCA, but they have kind of different prefixes. And what's very interesting is, in fact, we found that each of these motifs do have validation from a variety of independent previous experiments. So this is the main Nanog motif, which has the TCA. And then you have this one that has the GGAATT, and this one right here that has the AATGGC. I won't go too much into this, but in the paper, we showed that there's pretty strong evidence from crystal structure of monomeric Nanog binding to DNA, that at least this portion of the motif definitely is being utilized and is in contact with the protein. And there is also some evidence that from an EMSA assay that this flank is also bound by the TF. So we are learning different versions of the Nanog motif with slightly different footprints, with one main shared footprint and a bunch of others that are slightly different. Okay, so that's how we find the motifs and the motif instances. Our ultimate goal was to actually decipher motif syntax. So this is like a prerequisite in some sense. We first decipher the motifs. We find, as we see, there's quite a diversity of motifs. We identify the instances with high precision. And so now we move on to the arrangements, the syntax. So the first thing we wanted to check for is strict spacing syntax. So there has been various hypotheses about how transcription factors arrange themselves. And we want to see if there is evidence for strict syntax. By that, we mean very explicit, strict spacing constraints. And so we just ranked combinations of motifs based on strict spacing. And we looked at the frequencies in the genome. And we noticed that there's generally a depletion for strict syntax, except for 2 cases. So one case is where you have composite motifs through actual heterodimers. So for example, Nanog and Nanog alt often form a heterodimer. You often see, obviously, Oct4 form a heterodimer. You see Oct4 with Nanog sometimes forming a heterodimer, 8 base pairs. But in most other cases, these larger, we also found a bunch of strict spacing constraints amongst motifs like 46 base pairs, 68 base pairs. And if we started figuring out where these fall in the genome, pretty much all of them actually fall within retrotransposons. So we can essentially take strict spacing constraints and just bin them into two categories. One is composite motifs from actual heterodimers, and the other is basically artifacts of, not artifacts, but effects of background sequence. So retrotransposon, as you know, they have exactly the same sequence at multiple positions in the genome. The TFs are in fact binding those positions, and so you can get sort of a fake signal of strict spacing, which is actually simply an effect of the retrotransposon background sequence. But what's nice is even within these retrotransposons, we can take each of the transcription factors and really dissect their precise nucleotides within the retrotransposon that are bound by the TF of interest. So you can see KLF4, Nanog, Sox2, Oct4. And this again highlights the difference between a position frequency matrix, because a position frequency matrix just shows the entire retrotransposon, right? Because the entire sequence is repeated hundreds or thousands of times in the genome. So if you just look at frequency, every base in the retrotransposon pretty much lights up based on how conserved it is. But you can see that the importance scores or the contribution weight matrix precisely highlights the nucleotides that are actually associated with the footprints. And so it's a very nice way of homing in on predictive nucleotides within the retrotransposons the TF is bound to. Okay, so that's about fixed spacing. So we'll drop fixed spacing from now on because we really don't believe that we really see genuine instances of fixed spacing beyond short range composite motifs. So we start looking for more fuzzy patterns of syntax of spacing. So one pattern that we see is for the Nanog motif, if we look at the flanking sequences, if you look at the position frequency matrix, you don't see anything. You just see like the motif popping up, the TCA with that little thing at the side. But if you look at the contribution weight matrix, you see these really beautiful plumes of TAAT sequence, exactly 10.5 base pairs apart, which is a helical turn of DNA. And so one way to visualize this is you can actually take all the instances of the Nanog motif, which are the rows in this plot, and we're just plotting a heat map of the importance scores of all the nucleotides, and you can see these really nice bands of importance kind of radially dropping out exponentially with the 10.5 base pair frequency. And what's interesting is there has been previous evidence from other homeobox proteins that they can in fact bind nucleosomal DNA in these kinds of 10.5 base pair periodic sequences. So we think that the simplest explanation for this kind of broad 10.5 base pair binding preference for Nanog is that it binds potentially in the presence of nucleosomes. And we have some evidence from chemical mapping data. It basically binds on the same side of the helix, so that is accessible only from the solvent side. Okay. What's very interesting is all the motifs that contribute to Nanog binding, in fact, have this 10.5 base pair periodic flanking pattern. So the Oct-Sox motif, right, which obviously directly recruits Oct4 and Sox2, but indirectly affects Nanog, when it is in the Nanog, contributing to Nanog binding, it does show a 10.5 base pair periodic signal. The same motif, when you consider it with respect to Oct4, that sequence doesn't appear. The importance of these 10.5 base pair TAAT sequences disappears. So this periodic frequency that we're seeing in the flanks is a very specific property of Nanog. This allows us to actually make, in some sense, some approximate claims that the frequencies we're seeing is not some artifact of the exo nuclease, like pausing at certain positions, because if that was the case, we would see it for all the TFs. But this is very, very specific for Nanog. And as I said before, there is some evidence that homeoboxes can bind nucleosome DNA in these kinds of configurations. So what we decided to look at next is we wanted to look at preferred spacing between Nanog and other TFs, including itself. Because one hypothesis we had is this 10.5 base pair periodic flanking sequence that we see may also be reflected in the actual arrangements of the Nanog motifs next to each other as well as Nanog with other transcription factors. And in fact, that's exactly what we see. If we just take combinations of Nanog with itself, so whenever we see two pairs of predictive Nanog motifs next to each other in a sequence, and we just sort of plot a histogram of how often we see different spacings, we don't see evidence of strict spacing, but we see very nice evidence of soft spacing constraints. You can see that there is a preferred spacing, again, 10.5 base pairs that exponentially decays up to about 100, 150 base pairs. And what's interesting is this can only be seen when you use the CWMs and use them to scan the deep lift profiles. If you just take regular position weight matrices and scan these same sequences for the same motif, you actually do not see the signal as strongly because, again, it's very important for the neural network to highlight the predictive nucleotides to get to this kind of really clean periodic pattern. And we can use other methods as well, like checksmix, which also try to use footprinting patterns to decipher motif instances. But even in that case, we don't really see it very clearly. So this is a distinct advantage of using the neural network, the CWM representation, and the deep lift profiles to really highlight, get very clean signal for this 10.5 base pair periodicity. What's cool is we also see, just as we saw this this flanking periodicity for all the motifs that contribute to Nanog. Similarly, we look at heterodimeric spacing constraints between Nanog and other motifs. We actually again see 10.5 base pair periodicity. It's weaker than Nanog with itself, but it's still pretty strong. Now you might say, how is the model learning this? It must be in the data, right? It must be in the profiles. So if you just go back to the profiles and you align them based on these sequences, you in fact see that if you just look at the raw read counts from the chip nexus data, you can see it. But you can see it once you know what to look for. And that's the key. So this is actually just the raw data. This is just the read counts. You can see that there is an, again, preference of amplification in the read counts every 10.5 base pairs. This is for Nanog, this is for Sox2 and Nanog, this is for Oct4, Sox2 and Nanog. So obviously the model's not doing magic. It's homing in on this pattern and projecting it back down onto the sequence patterns that are predictive of these patterns that you see in the reads itself. Okay, so that's spacing constraints. Now let's address the last part, which is cooperativity. By cooperativity, what we mean is the influence of one motif or binding of one transcription factor on a binding of another transcription factor, okay? And so we developed two methods to address this, this question using basically the model as an in-silico oracle. So the key thing is if you're really trying to figure cooperative binding of transcription factors, you can't just conclude that from looking at spacing constraints and so forth. You really need perturbation experiments. You need to perturb one motif and see what effect it has on another TF. You need to perturb another motif, see what effect it has on another TF. And then you perturb both and you see what effect they jointly have, right? Typically, these are done using experiments, like you would design synthetic sequences and you would do binding experiments in vitro, or you would do an MPRA or some such. Well, if our model is as predictive as an experiment, at least from our evaluations, the footprint profiles are extremely accurate at the level of concordance with replicates, we can then use the model like an oracle, and we can do all these kinds of experiments in silico, just as you would do them in the cell, right? Using synthetic sequences or CRISPR experiments. So one approach we take is we design synthetic sequences. We dump motifs in those sequences. And then we can play with various properties, see spacing, orientation, and so forth, and use a model to predict what would happen to binding. The other approach is to take genomic sequences that are actually in the genome, which have an existing configuration, and then perform various kinds of mutation experiments where you mutate motifs in combinations or individually, and use a model to predict what would happen to binding at base resolution. And so we call this in-silico biochemistry and in-silico genetics. So I'll show you two examples of this real quick. So this is a synthetic experiment. We are trying to understand the spacing constraints and the effects that these spacing constraints have on Oct4 and Nanog binding with respect to the Oct Sox motif, as shown right here, and the Nanog motif shown right here. So what we'll do is we'll take a synthetic sequence, some random sequence, which we verify is benign, does not have much of an effect on binding of either of the two TFs. We'll insert Nanog at this position, the Nanog motif, and we'll insert the Oct4 motif at this position. Then we will start moving the Oct4 motif towards the Nanog motif, and the model will predict what happens to Nanog binding and Oct4 binding. And we can graph this, as shown here. And we can, of course, do this millions of times with various kinds of backgrounds to make sure that we're not like making any, we're not sensitive to the background sequence being used. And what you end up seeing is some really interesting behavior. So this is on the y-axis is the change in the footprint height as a function of distance between the two motifs. So the yellow curve is showing you the effect of moving the Oct4 motif towards a Nanog motif on Nanog binding, okay? And you see it has a massive impact. So first of all, like there's huge spikes in increase in footprint height when the Oct4 motif is nearby, and as you move it away, it drops. So there's an exponential decay in the effect of Oct4 motif on Nanog binding. But more interestingly, there is this very strong 10.5 base pair periodic cooperativity that you see again. If you invert the relationship and you see, does Nanog have an influence on Oct4 binding? The answer is no, it does not. So Oct4 kind of does its own thing, classic pioneer factor behavior. On the other hand, it is having a massive influence on Nanog, not just proximally, but all the way up to 150 base pairs. So that's a nucleosome range effect. So there's a short range effect mediated by helical periodicity of DNA and a nucleosomal effect that goes up to 150 base pairs. Now we can repeat the same experiment, but instead of this kind of synthetic sequences, we can do the same thing using sequences in the genome. And so here we will effectively do an in-silico CRISPR-like experiment where we take, again, this Oct4 enhancer, Oct4 and Nanog bind. You can see the Nanog motif is specifically predictive for Nanog binding, whereas the Oct4 motif is important for Oct4 as well as Nanog. And so we'll first delete the Oct4 motif. So remove it, or we'll replace it with random nucleotides. And you can see that the model predicts that as soon as you remove the Oct4 motif, Oct4 binding disappears and Nanog binding disappears. So Oct4 has a really strong effect on Nanog. On the other hand, if you delete the Nanog motif, you will notice that the Oct4 profile remains pretty much untouched. And the Nanog motif, the Nanog profile, in fact, doesn't disappear. It dramatically drops, but even after you remove its direct binding motif, just the Oct4 motif can in fact have a weak indirect recruitment effect on Nanog, okay? And we can again average this across all the instances in the genome, and we observe something very similar to what we saw from the from the synthetic experiment. We see that Oct4 has this really strong impact on Nanog with a 10.5 base pair periodicity, whereas the reverse is not true all the way up to when it comes to really nearby. Okay, so we have a nice sort of verification from the synthetic as well as the genomic sequence perturbation experiments of this really nice asymmetric relationship between Oct4 and Nanog, and also the fact that there's a soft helical periodic preference of spacing, which enhances cooperativity, and that extends all the way up to 150 base pairs, nucleosome range. And so we can do this for all kinds of combinations, and we will see some very interesting patterns. The classic behavior of Oct4 as a pioneer motif, super strong effects on pretty much everything except KLF4. KLF4 seems to kind of do its own thing. Not really strong effects on anybody and not impacted as much by anyone. And again, you see this, some TFs like Oct4 and even Sox2 can have cooperative effects all the way up to 150 base pairs. On the other hand, you can see Nanog having cooperativity with itself, but on a much shorter distance scale, right? So Nanog is not really having very long range, nucleosome range effects on itself or others. And so we can summarize this effectively as this kind of impact graph which shows how deletions of different motifs would have effects on different TFs. And you can again see that this behavior is extremely asymmetric. That is, some TFs have very strong effects. Some motifs have very strong effects on cooperative binding for all transcription factors, whereas other motifs don't have any effect but are impacted very strongly by everybody, like Nanog. And some of these, as you can see, can extend up to nucleosome range. So those are our predictions. Of course, we have to do some validation of these predictions. It's pretty hard to do large-scale CRISPR experiments and measure binding. So Julia's lab actually put in some Herculean efforts to do a handful of CRISPR mutations for specific entities, elements, and measure chip nexus genome wide. So I'll show you two examples. This is a region in the genome that's jointly bound by Nanog and Sox2. Here you can see the Sox2 motif. This is what the model predicts on the sequence. This is the observed. This is what you actually measure. You can see there's pretty nice agreement between the profiles at base resolution. Now what Julia's lab did is they introduced 2 nucleotide mutation in the Sox2 motif, and they measured Sox2 chip nexus again. And we also predicted what would happen through the model to Sox2 binding. So you can see the model predicts that if you mutate this motif, the Sox2 footprint disappears, the red profile, and there is a slight attenuation of Sox2 binding all the way up to here, right? And so if you actually do the actual experiment, you see the same thing. You see again attenuation of SOX2 binding at this position, complete attenuation and partial attenuation at this position. So that really showcases, again, verifies in some sense, not just the predictions of the model, but also this kind of longer range impact of SOX2 on itself. We can also look at the effects on Nanog, and you again see very interesting effects. You're again deleting the you're mutating the SOX2 motif, and you're measuring the effects on Nanog chip nexus. And you can again see there are pretty long range effects of the SOX2 motif on Nanog binding. And the model's predictions are very much in line with what you get from the experiment.
Anshul, I'm sorry to interrupt you. There's 4 minutes left, including a Q&A session. So just wanted to make sure you were aware of that.
Yeah, it's my last slide, last 2 slides. So we can do the same thing where we mutate the Nanog motif, and we see it has very little impact or no impact on SOX2 binding. Sorry, I missed something. And the Nanog motif does have a strong effect on Nanog binding, but it's mostly proximal. Okay, so this really validates the model's predictions. We also did large-scale validation. I'll skip this in the interest of time, but we can show that the BPNet representation can also be very accurately used to predict genome-wide changes in attack-seq profiles after Oct4 and Sox2 depletion of the transcription factor. And we also show that the model's representation is also very predictive of expression effects from massively parallel report assays. So I'll just end there and summarize. The BPNet can be used to map raw DNA sequence to base resolution profiles. We've applied this to Chip-exo nexus, and as you'll see in Alex's talk next, it also applies very nicely to chip-seq, attack-seq, DNA-seq. We have all these nice interpretation methods with the you can see the focus really is on deciphering higher order syntax and cooperativity. And finally, the models are really quite accurate, and they can predict the effects of motif perturbations at base resolution, differential chromatin accessibility up to TF knockdown, and report expression activity. I'll skip this. I'll also just quickly highlight, we've released a repository called KIPI for machine learning models for genomics, jointly with Ollie and Julian. And you should feel free to go here and contribute your models, as well as use them for predictions. And again, I'll just end by thanking everybody who helped make this work possible. Thanks.
Thank you so much for the wonderful presentation. So we're a little bit, we have 2 minutes in this session, but because there's a 20-minute break, I'll suggest that we take a couple more questions, and then the remainder of the questions, maybe Anshul can answer them by typing in the QA kind of panel. So I'll start from the top question. This is from Bill Noble. He asked, can you explain why the 10.5 BP Nanog periodicity shows up for the CWM scores, but not from the information content of standard PWM?
Yeah, I think that's a great question. So I think partially the reason is that the sequence, those 10.5 base pair periodicities are present in the sequence, but they are relatively weak signals. So if you just, even if you get the motif position exactly right, if you just take the frequencies of nucleotides, then ACs, Gs, and Ts all look alike. But if you look at the predictive importance, the neural network specifically says, I like those ATs. And so it amplifies the frequencies of the ATs by reweighting them higher. And that's the reason you don't see them if you just take frequency representations, but you do see them if you use the importance scores, because the ATs are equally frequent as the CGs, but the neural network increases their importance. And so the importance scores actually light up.
Okay, thanks. So second question, also by Bill Noble, that's been voted up. How expensive is it to train BPNet?
Actually, it's remarkably fast. You can train a BPNet model on 4 transcription factors in about 20 minutes on a single GPU. So it's actually really fast. It's in fact, you can train it genome wide, or you can train it only on the peak regions. And of course, training on the peak regions is even faster. And in fact, we see no reductions in any of the interpretations by just focusing on the bound regions. So it's pretty efficient.
Great. So the third question, I think this refers to the way you were binarizing your data for predictions.
Mm-hmm.
Correct me if I'm wrong, Isawarna. So to define positive spike positions during the evaluation, what threshold is used? Have you tried evaluating at different thresholds?
Yes. So the threshold is based on the signal to noise ratio in the data set, and it's determined on the training set. So we don't really, again, touch the test set. It's only determined based on signal to noise ratios in the training set. Then we transfer that same threshold to the test set. That's where we get the labels, and then we evaluate. We have tried other thresholding mechanisms, and across the board, basically, these values might change a bit, but the relative performance with respect to replicates and so forth remains pretty much the same.
Next question from Jan. This goes back to the 10.5 base pair pattern. So does this mean that the 10.5 base pair pattern only from CWM, because the information content has been encoded in the deeper layers of BPNet?
Yeah, I can't comment on whether the information is in the deeper layers or not, but I think one of the things we do is, yes, we use the entire network and propagate all the neurons, predictions or importance back onto the basis, right? Individual nucleotides. And so, yes, the whole network is actually voting on every nucleotide, and it is finding the ATs to be important. And that's why it's basically highlighting them up.
Thank you. Next question is, next question I think you also answered is about binarizing the profile. So I'll just skip to the question after that. So in the plot where you're showing AUPRC graphs, what's the orange? Can you please explain that?
So the orange, I'm assuming you mean the yellow, I guess, the AUPRC graph. So the yellow line is corresponding to if you took one replicate of an experiment and use it to predict another replicate. So it's basically measuring concordance between replicates, right? So you use one replicate like as if it's making predictions about the other replicate, and you use it to predict the footprints from the other replicate. That's the yellow line. That's the AUPRC of, in some sense, it's a measure of the concordance between replicates for footprint precision.
Great, thank you. So maybe for interest of time, I'll just ask one more question, and then if Anshul is kind enough to answer the rest by typing, so that we could have a 20-minute break. But so the last question that I'll ask is, why do you set up the loss function in this way? I guess the composite loss. What would happen if you just one of the two losses?
Yeah, so if you set it up as one loss, then you get basically one prediction. And what would happen is obviously the model's performance collectively on the profile and the counts is not that great. So first of all, you wouldn't know that the model is able to make predictions on the profiles as accurately. And secondly, the multinomial loss, in fact, is a very nice way to capture the sparsity of the data, because you have an underlying probability distribution across the sequence, right? And then depending on the total number of reads that I throw on the 1,000 base pairs, I can get different number of read counts. And if I have very few reads thrown at it, I will see a lot of zeros. Those are obviously not real zeros, right? So the multinomial captures the sparsity properties really quite nicely. We have tried also using mean squared error and other kinds of, Poisson loss per base, but it doesn't capture the dependency that you see. The multinomial really distributes the reads across the entire sequence. And so it captures some nice properties of how if you increase reads at one position, how it reduce reads at the other. It captures some of that dependency quite nicely. And so we find that it is actually quite powerful, and it's a very nice way to separate the total counts from the profile shapes in terms of outputs as well. And I didn't show you in the talk today, but you do in fact find motifs that can have contributions to the total counts, but not to the profiles necessarily. So they kind of increase the occupancy, but they don't necessarily change the shapes of the footprints. So you can decouple these two properties really quite nicely. And that's another advantage of the sort of the separating these two components out. There might be other loss functions that would work just as well, but we find that the multinomial is actually quite nice.
Cool, thank you. And maybe just as a privilege of being the moderator, I'll ask you the last question relating to this. For the composite loss, I guess, is there some hyperparameter that allows you to balance the two types of errors, and how sensitive is the model to that, and how do you adjust for it?
Yeah, great question. So in the paper, we actually derive a way to, in a somewhat principled way, to set that weighting parameter between the two losses. And what we find is if you are, in general, it's a function that weighting parameter is a function of the number of reads. And we have a way of setting it. It's usually number of reads by 10. It works very well out of the box in most cases. When you have very deep data or very shallow data, you might have to flip around with those, but we've applied this to lots of Chip-seq and Chip-exo experiments, and usually that n by 10 ratio, which we kind of derive based on decoupling the two losses and relating it to a Poisson loss, it works quite well in practice. So you don't have to really hand tune it a ton, but it is an important parameter, so you can't just randomly set it. But we have a default that tends to work quite nicely.